Good morning all. Today we will do a practicals on qualitative analysis of proteins. The aim of this experiment is to detect the presence or absence of proteins in the given samples. Proteins are complex organic molecules containing carbon, hydrogen, oxygen and nitrogen. They are macromolecules made of long polypeptide chains. The unit of a protein is amino acid. We'll perform some simple tests today to detect the presence of proteins. The tests that we are going to do are Biuret test, Millen's test, xanthoprotic test and Ninhydrin test. We'll do the practicals now. I've taken a sample in which we have to detect the proteins. Reagents, Biuret reagent, Millen's reagent, concentrated nitric acid, ammonium hydroxide and Ninhydrin reagent. A few clean glass test tubes, spirit lamp, matchbox and a test tube holder. We'll do the tests now. First test is Biuret test. In a clean glass test tube, take about 2 ml of the sample. Add Biuret reagent. You can see Biuret is light blue in color. As you add, keep shaking the test tube and shaking the contents. The solution turns purple. So this indicates the presence of proteins. Second test is the Millen's test. In a test tube, we take 2 ml of the sample, add Millen's reagent. As you add, you can see the formation of white color. Mix the contents thoroughly. You get a white precipitate. We have to heat this precipitate now. We'll keep the sample and the reagents a little away. So hold the test tube with a test tube holder and heat the solution. So as the solution heats, you can see the precipitate turns orange in color or reddish brown. This indicates the presence of proteins. Next we will do the xanthoprotic test. Take about 2 ml of sample in a clean test tube. Add concentrated nitric acid. So hold the test tube in a slanting position and add the acid along the sides of the test tube. So you can see a precipitate that has formed. We have to heat this precipitate. Hold the test tubes tightly with the test tube holder and start heating. Be careful. Face the test tube away from you. You can see as it heats, the color changes. The color changes into yellow. We have to cool this and then add strong ammonium hydroxide. You can see the solution has turned yellow. So after the solution cools, you can hold it under the tap, running water to cool it. Add ammonium hydroxide. So the yellow color turns into orange, indicating the presence of protein. The last test we are going to do today is the Ninhydrin test. Into a clean test tube, take about 2 ml of sample. Add a few drops of Ninhydrin reagent. Mix the contents thoroughly and heat. Hold the test tube tightly with a test tube holder and heat the solution. As it boils, you can see the appearance of a blue color. This indicates the presence of proteins. I have kept the test results here. For BioRed, purple coloration. For Millen's, reddish brown precipitate. Xanthoprotic test, orange coloration or precipitate. And for Ninhydrin, blue coloration. All these tests indicate the presence of proteins in a given sample. I have given the procedure here: BioRed test, Millen's test, Xanthoprotic test and Ninhydrin test. BioRed and Ninhydrin are general test. Most of the sample will answer for it. BioRed is a general test for peptide linkage, whereas Ninhydrin is a test for amino acid. Millen's and Xanthoprotic indicate the presence of certain amino acids like tyrosine, tryptophan and phenylalanine. After performing the experiments, you have to tabulate your results. So depending on how many of our samples have been given for exam, suppose if there are 2 samples, take 2 test tubes, label them sample A and sample B. If 3 samples are given, take 3 test tubes and label them sample A, B and C. Then perform all the 4 tests in each sample and tabulate your results whether it is positive or negative. If a blank sample is given, then it will give a negative result for all the tests. After tabulating, you have to give the result as to which sample contains protein and which sample does not have protein.